All right, our speaker today is Brad Schaefer, distinguished professor in the Department of Ecology and Evolutionary Biology at UCLA. Brad is also director of the Lacrette Center for California Conservation Science. I first met Brad a long time ago, probably I think 1975, when he transferred to Berkeley from UC Santa Barbara. We had an outstanding group of undergrads in MVZ at the time, many very successful in subsequent years, including, in addition to Brad, such people as Jim Henkin, Jim Griesemer, Paul Elias, and Mimi Zeldich. Brad was interested in turtles, but he is out of luck with me. Nevertheless, we have been good friends ever since. His first research project dealt with predation rates on salamanders from my Guatemalan study site. This led to his first nice publication. Brad did his graduate work at the University of Chicago with Steve Arnold, an ex-Miller fellow I had sponsored, and who Brad knew during Steve's first job at Santa Barbara. Brad worked on tiger salamanders, doing much of his Alzheimer's work in the MVZ genetics lab. He received his PhD in 1982. Following postdocs at Wisconsin and Chicago, Brad joined the faculty at UC Irvine, and after a couple of years transferred to UC Davis, where he remained until 2012, when he accepted his current challenge at UCLA. At Davis, he built a powerhouse program in conservation biology and in evolutionary and ecological genetics. He continued long-range studies of tiger salamanders and laid a heavy emphasis on turtles, but also much else. He was notably successful in attracting outstanding graduate students, including our own Ian Wang, and postdocs. Lots of undergraduates from his lab went on to successful careers elsewhere. His program at UCLA is equally successful. Brad is a prolific researcher who's published much important work. At present, he has 2 impressive papers on turtle evolution, both about to appear in different prominent publications. Brad is the motivator and lead PI of the $12 million California Conservation Genomics project, which involves lots of MVZ people. And that will be the primary focus of his presentation today. Welcome, Brad, and take it away.
Thank you, Dave. Thank you very much. Do I have screen share capabilities?
Yes, you should.
I should. Okay. Then let me do that, and let us hope my computer continues to be at least reasonably good. Are we good to go?
Yes.
Yes. Okay, great. Well, Dave, first of all, thank you. And I know I'm going to have to cut this short, but one piece of this before I get into the California Conservation Genomics Project, and I list on this cover page our executive committee, the people who have really helped me formulate and grow this project, including very much Ian from your department or from your campus. Here's that paper that Dave was referring to. It's the first paper ever published. Predation pressure along the Dave's MVZ Guatemala transect. And I just want to make a point. I want to make a point both to the faculty or soon to be faculty who are on this call, as well as to the undergrads. So this is a project that started when I was in the MVZ. I was always a herpetology nut. I always loved turtles, as Dave said, but also other herps. So that I didn't pick up at MVZ. What I did pick up was a research project and learning what research was about. This wasn't for a class or anything else. And it started in about probably 1976, something like that. And it was something very much done with Dave and also with Ray Huey, who was a postdoc, Miller postdoc at the time. And my point on this is that, it's been close to 45 years since this started. During that time, Dave and I have published been co-authors on one relatively minor paper together. Otherwise, we've never published anything together. Ray and I have published a couple of things, but not much. But both of them, as well as lots of other Berkeley faculty, became sort of lifelong colleagues of mine because of doing work in the MVZ. And that's something that I picked up very much as an important theme in my own work. When I got a job and moved, my first sort of major job was at UC Davis. And, you know, a bunch of folks who are undergrads in my lab, people who you might know, Robert Fisher, Duncan Urschick, Chris Austin, Kat Darst, who's now head of listing and recovery with the Fish and Wildlife Service, Angela Pico, who's also with the service, lots of folks, lots of undergrads. And they became sort of lifelong colleagues and lifelong, you know, folks who I work with constantly. And it's something I picked up from Dave at Berkeley. And I just want to say, you know, if you're an undergrad, make that connection. If you're a faculty member, respect those undergrads because they can turn into some of the most wonderful relationships of your entire career. They certainly have with me. So just a shout out there. Okay, I know we're quite short on time because of computer screw up. So let me jump right in here. And I think there's probably some stuff that I'm going to skip, which we may not need to have. But what I want to do in this talk really is talk to you about this project, the CCGP, the California Conservation Genomics Project. It's a big, ambitious project that we got off the ground about a year ago. I think it's relevant to give an update and a little bit of perspective on the project to folks at Berkeley, since Berkeley is the biggest recipient of this project. More money from it is going to Berkeley than any other campus. And of that, the vast majority of it is going to MVZ or IB folks. So it's a very Berkeley relevant project. And in many ways, it reflects the, you know, sort of culmination of work that I learned about when I was an undergrad at Berkeley and have sort of kept as a theme through my entire career, which is really using sort of landscape genetics, landscape genomics techniques, in my case, to answer issues and questions in conservation. So I think what I'm going to do is, as I said, skip through a few things so that we can really get down to the bottom of this talk, which is, you know, the CCGP, the California Conservation Genomics Project. And hopefully we all know that genetics is important for conservation. If we don't, we should, because it is. And I would actually argue, cutting to the chase, that there's sort of two reasons why it's important. The first is what neutral variation tells us, which I think right now these days is viewed as a little less exciting than what genes that matter tell us. So, you know, neutral variation tells us about population structure, gene flow, hybridization, demography, those sort of standard things that we've all worked on for years. But I would argue for conservation, those inferences from neutral variation are actually the most important ones. I mean, they're both important, but the ones that garner the most respect in the sort of agency world, the ones that they care about the most are things like population structure, identifying management units, those kind of very basic, very standard things. And they are critical for the relationship between conservation and genetics. The way we've done that in the past is using sort of relatively simple markers and simple data sets to try and understand particular issues about particular landscapes with particular species. And I've got a couple of examples that are from, you know, one from a very nice study using microsatellites that was done in the Santa Monica Mountains to ask whether what sort of the impact of the 101 freeway as it runs east-west across the Santa Monicas has been important. And the bottom line on it is that looking at, side blotch lizards with sort of a sketchy set of localities on either side of the 101. And we can sort of see that there's sort of a break in population genetic structure that might align with the 101, and that's kind of interesting. We use a different sampling design, a different species here at Rentit. And, again, we sort of see what maybe we could say has something to do with a barrier to gene flow across the 101. Bobcats, maybe the same thing. Actually, bobcats look a little more like they really do line up with the freeway, except in the West where they don't. And we can sort of cobble those things together and try and make some inferences across a few species with different sampling designs, different sets of markers, different power to try to say something that's relevant to conservation. And that's kind of one strategy that's been used very successfully. Another form of business as usual is what a lot of us are doing right now. A lot of people in the MVZ, everybody in my lab. And this is an example paper that was accepted this morning, actually, on spadefoot toads, candidate for listing under the US Endangered Species Act, work that we've been doing to look at them. And again, this is a very kind of standard sampling design that many of us might use. We sample across the entire range of Western spadefoot, Spea hammondii, across its range. We do things like PCAs, we do clustering analyses, we do that based on 10 or 20 or 30,000 SNP markers, and we make some inferences. There's two different groups in the north and the south, or if we kind of increase the K down towards the bottom, we can see some level of differentiation across even individual ponds on very small landscapes. And we use that to make inferences, and we learn something about the genetic structure and landscape genetics of that species on that landscape with that set of markers. And that's great. It's important, it's useful. It certainly is important for both conservation and population biology. We can also ask, and I think what I'm going to do is just skip this part because we are pretty short on time. It's a really great meta-analysis where we are in the process of, with a group of undergrads from UCLA, going through the published literature as best as we can on all studies of genetic variation published in California ever on natural populations. It's whatever we said, it's 4,687 papers, 550 species, and we're trying to sort of summarize that. And it tells us some interesting stuff, but I think we're, in the name of time, we're going to sort of take a pass on that right now. So what are the, whether it's RADseq on spadefoots or looking at a meta-analysis for the literature, the big problems that I've always seen with this come in a few flavors. One of them, the big one, is data combinability. It's hard to combine different studies. You can sort of use meta-analysis techniques to do a workaround on that, but they're never terribly satisfying. And we never, at least to me, we never feel like we can really apply them equally to landscapes that we're interested in. There's problems. Part of that combinability issue is data consistency. One study is using microsatellites, one's using RAD, one's using target capture. Their coverage of the genome is at different depths and different levels. It makes it very hard to, say, map out heterozygosity across the state. It's hard to add new data. That's certainly true with things like RAD, where you can't sort of combine studies terribly well because they're using different enzymes, different markers. There's a problem with non-homology of markers, and we do lack information from the entire genome. So couple of years ago, I sat down with a colleague of mine named Mark Gold, and we had been trying to work on this for a number of years. And we said, how can we do this better? And our approach to that is what the CCGP is. So rather than looking at my spadefoots or my tiger salamanders or John Richmond's blunt-nosed leopard lizards or Steve Beisinger's black rails, sort of in isolation and independently, we said, what if we could look at a whole pile of species, hundreds of species across the state of California, sampled in a relatively consistent way, using the same kind of data on all of them, and in our case, whole genome data. So everything done at the whole genome resequencing level, with all of those whole genomes mapped onto really high quality reference genomes, and done in a consistent way so the data are combinable, so that we can really generate a picture of genetic variation across the state of California. Wouldn't that be amazing? And we thought it would be. And it turned out that with a little work, we were able to convince a member of the state legislature to promote this as an important tool that we could use to help sort of guide certain conservation actions in the state of California. And he put it on the state budget as a line item in the 2019 budget. That line item was funded, and the CCGP, or California Conservation Genomics Project, was born. So at a glance, what is it? It's currently sitting at about 225 species that are being covered. They fall into 130 projects. So a project might be one species or several very closely related species that sort of together form a unit. So, right, okay. The strategy is for each one of those projects, we put together one reference genome, and that's going to be a really good reference genome. For those of you who care about this stuff, that's PacBio long read sequencing using OmniC high C assembler to put those long read assemblies into very long contigs, sort of chromosome level contigs. Do one of those and have RNA-seq for annotating that genome. One reference genome for every project. Okay, so every set of very closely related congeneric species. In addition, for each one of those, have 150 individuals, and I've gone through this with many of you on this call, 150 fully resequenced individuals per project that are resequenced at the roughly 10x coverage for their entire genome using Illumina short read sequencing, which is then mapped back onto those reference genomes to give us 150 fully resequenced individuals. We chose 10x coverage because you can pretty well call hets if you're up around 10x coverage. So what does that, do the arithmetic, and 150 times 130 is getting close to 20,000 individuals resequenced at their whole genome level. Some of those have very small genomes, they're a couple of hundred megabases. Some of them have genomes that are 5 gigabases, and we're just trying to get all of that done. Some of the features of the project that I've been battling with or trying to set up over the last year, we decided to set up a dedicated core facility at UCLA that would do at or below cost DNA isolation, library preparations, and pooling for anybody who wanted to take advantage of that. We're shooting for, as best we can, uniform genomic coverage at the 10x level, like I said, and then we're setting up sort of common analysis pipelines that all of those data will flow through, as well as, of course, the data going back to the PIs to do whatever they want with it. Okay. What do we hope to accomplish? Well, we hope to accomplish, this. taxonomic ecological coverage for the state replicated across many species. And why do we want that? What are we going to do with it? Well, in my mind, the kind of single most important thing to do with that data set is to create sort of individual heat maps of genetic variation for each of those species or each of those projects across the state. And that's for marine taxa, for terrestrial taxa. And then be able to stack those up to ask the question, where do we see regions that are sort of hot spots and cold spots for genetic variation across the state? Is it true that the Tehachapi's are sort of a universal hotspot for genetic variation? Is it true that the top of the Sierras is a universal cold spot? What about the Central Valley? What about the Trinity Alps? And across lots of species that cover lots of ecological types, under the assumption that those hotspots are our best kind of guess for climate resiliency and therefore are buffering our biodiversity against climate change, at least to some extent. Are those hotspots protected and protected adequately? Are there corridors that connect them so the genes that are important in them can move around between those hotspots? And use that to identify lands that groups like the Wildlife Conservation Board, which is a major player in California conservation, can use to identify land that's important for acquisition or for additional protection. So the team, we've got 72 principal investigators from all 10 UC campuses. We have an executive committee, and we have a very large number of co-PIs, and the California Department of Food and Agriculture is also a player in this and is contributing some insights and work on pest species. The funding, as Dave mentioned, it's a total of $12 million that the state has dedicated to this over a 3-year project. About 10 of that goes directly out to the labs. So we've pushed out about 8.5 or 9 million so far to all of the labs. So most of that funding is not staying at UCLA, but we are administering it. And again, the goal is that what that funding is buying us is a very large body of consistent data. And the way we're sort of ensuring that consistency to some extent, and we're working with many of you right now on this, is that all the reference genomes are just being done by us, or by, the hi-fi work is all being done over at Davis. The OmniSeq work is all being done at Santa Cruz. The RNA work is being done down here at UCLA. But we're just making that really consistent by saying we don't want the PIs to do it. We're just going to do it for you. All the resequencing is done to sort of maximize coverage across the state, and Ian has been a real player in helping make sure that that's consistent and successful. And then most of the projects have some conservation sensitive species in them. So let's see how we're doing on time. Not great. Okay, so how are we doing? What I'm mapping here is, again, this was one of Ian's brilliant insights, is that for every project, we asked PIs to say, where are your samples coming from? And we put those all into a standardized spreadsheet format. And so we can at least look at how the coverage looks. So how's our coverage at this point, including specimens that are already in hand or that people promise they will have in hand? And I would say it's pretty good. There's our marine coverage up and down the coast. So each dot is an individual here. Many times they're stacked up. Plants, we have very good coverage, except in the Central Valley and especially in the San Joaquin, where there's just not much habitat left. You'll see kind of consistently that the deserts of Southeast and Northeastern California or Eastern California are somewhat underrepresented, but really good on plants. If we throw in vertebrates, those same holes are still there, but we have very good coverage on vertebrates, and even terrestrial invertebrates are coming together really quite nicely. I think we have really good, it's not as as dense, but really good spatial coverage across the state. One of the things we've really been wanting to do is make sure we have representation across the 18 ecoregions that we're using for the state. And this just shows you how many species are in each of those 18. And again, you can see the coast, the coastal regions are somewhat overrepresented. The deserts, especially down in southeastern California and like up in the Modoc Plateau and stuff, are somewhat underrepresented, partly because they're lower diversity, partly because there's just less, research interest up there. But overall, I think it's pretty good. I feel pretty good about it. So progress on the project to date. The funds are mostly distributed. That's been a big one. Figuring out how to do that fairly and transparently has been taken up most of my life for the last year. That's pretty well done. We have about 45 of the 130 reference genomes are in play, meaning the material has been sent to the facilities, and we're gonna, we're starting to work our way through those. The goal is to have all the reference genomes and about half of the resequencing done by July 1st of next year, and all of the data in a year from then. We'll see if we can do that or not. A couple things I'll share with you quickly, and then we can call it quits on an abbreviated version of this talk. These are a couple of maps that just show our sampling. So on the left in orange, you can see all of the samples from across all those projects that are already in hand. And so if no one does another minute of field work, at least at the level of coverage, we're in pretty good shape for the state. The sampling to be done, which is about a third of the total, has a very similar distribution. And if we put those two together, then we get this kind of a map. But I think the point is that we already have, or the PIs already have, about 2/3 of the samples in hand. And that feels kind of very satisfying and very good. One of the things we've done that Ryan Harrigan, one of the members of our team down here at UCLA, put together is to try to take all of our sampling and get in touch with PIs so that when those 4,000 plus samples need to be collected, perhaps people can actually work together. It saves gasoline, it saves time. It might lead to some efficiencies from a big project like this. So here's all of the sites. And then what Ryan did is put sort of 5-kilometer circles around each site. And what we've done is with this kind of data, and everybody should have gotten one of these, at least all the PIs should have, is a list of all of your sites and the 5 closest sites to each of your sites that you still need to collect, and who the PIs are that are going to sites within 5 kilometers of your sites. So the idea is that if Michael and I both need to go to the same place, and he needs to get a mouse and I need to get a snake, maybe we can actually work together and sort of gain some efficiencies on that. So that just went out this week, I believe, to all of the PIs. So that's something we're trying to do. In terms of the reference genome part of it, here's where we're at. This is as of yesterday, I believe. So we have about 46 of the species have high molecular weight tissue that has been sent in to UC Davis. About 42, mostly the same, a subset of the 46, have also been sent over to UC Santa Cruz for OmniC sequencing for stitching those long-read assemblies together. About half of those material has come down here to UCLA for RNA-seq and so on. How those are looking, here's just, we've got a couple, we have 4 now that are where at least the hi-fi PacBio sequencing has been done. And this is from a black abalone that somebody at Davis is the lead on, I believe. And if you just look at the statistics on this, they're really pretty amazing, right? So, I mean, to my mind, the biggest statistic that many of us like to think about is the contig N50. It's 14 megabases. That's pretty astonishing. You know, I mean, it used to be that we'd be happy with a 10th of that, or, you know, even a 20th of that. It almost means you don't need to do the high C further assembly. I mean, those are such big contigs. You know, the longest contig just from the sequencing is 56, 57 megabases. It's really pretty astonishing. And I think this reflects the fact that the hi-fi sequencing from PacBio on their new machine, their new Sequel 2 or 2E, is really pretty great. Those BUSCO scores are quite amazing. And hopefully we will have this level of reference genome for every one of those 130 species. And we'll have it, I hope, by next July, assuming people get their tissues in. So that's pretty cool. 3 cool things we're doing. It's already 1 o'clock. My guess is that people have to leave. We'll just go through this real quickly. We want to make these heat maps of variation. I can't show you any because those aren't done yet. This is an example from some work we did on foothill yellow-legged frogs using RADseq. You know, but we want hundreds of these kinds of maps that we can use to look at areas where genetic turnover is the greatest, where genetic diversity is the highest, that sort of thing. There are some very important forensic applications. This happens to be one from Western pond turtle, one of the species that Ian and I are doing. And there's a population that showed up out at Edwards Air Force Base. No one has a clue as to what in the world those damn turtles are doing out there. Don't worry about the inguinal scute. But there's Edwards. We should be able to use this in a very, very straightforward way to say, are those animals from Edwards release animals perhaps that came from Emys marmorata in the Sierras, perhaps from coastal Emys pallida, or perhaps there are a couple of natural populations of Western pond turtles out in the Mojave River. Maybe they really are native. You know, really, maybe they weren't introduced. Maybe just no one ever noticed them out at Edwards before. I doubt that, but it's possible. And so we can do that. There's lots of applications for poaching with things like abalone or Dungeness crab, all of which are species in the mix that I think will help the California Department of Fish and Wildlife in a lot of their forensics work. And finally, this is, I think, just about the last thing I'll say. Many people have asked me, why in the world are we doing this with whole genome data? We don't need whole genome data. You know, RADseq will tell these stories as well as whole genomes. It's easier, it's cheaper. Well, I don't know if it's easier, but it's cheaper. And I mean, people like Michael, who care about genes important in adaptation, you know, or Bob Wayne down here, certainly would say, of course you do it at the whole genome level. But even for more sort of standard studies using neutral variation, the point is that we really want to make this a lasting resource, and a resource that'll work no matter what markers people are using and no matter how the field changes in the future. We really want it to be a baseline reference study for genomic variation for the state in the future. And one of the things about it is that if you do this kind of thing, if you make these reference maps using, say, RADseq, which many of us use right now, it means that in the future, if new data come in, or in the past, if there were old sequence data that maybe told an interesting story, you can't use those new data. The only thing you can use is RADseq data that uses the exact same restriction enzymes and is done in exactly the same way with the same size selection on your pip and prep, or whatever it is. You can only compare data sets that are done in exactly the same way. And really importantly, you can't compare old data sets. If somebody's got some old Sanger sequencing, you just can't put that in the context of this RADseq. Whereas if everything is whole genome, it can, and there's a good reference for every species, everything can be mapped back onto that. I mean, Alzheimer's data can't, but anything that's at the sequence level can be mapped back on and can be put in the context of the genomic work that's been done across the whole state. And to my mind, and to the mind of everybody on our executive committee, that just makes all the difference. And it's not that expensive these days when you're paying $7 a gigabase for sequence data. The resequencing really doesn't amount to all that much money. And the flexibility, especially looking to the future, with having that as the reference set is really quite extraordinary. So that's why we decided to do that. So there's the goal. I've already said that. We have a website that is up already. If you just Google CCGP UCLA, you'll pick it up. We're completely revamping that website as we speak, trying to make it a sort of broad and inclusive website that has sections for scientists like people on this call, sections for policy and decision makers, and sections for what we call the curious conservationists, folks who are just curious about why this is interesting and important and want to learn more about it. We have the opportunities, especially as the data start rolling in, to have web-based displays of those data. This is just one. I'm not going to, thank God, try to log on and do this live. But for example, we can do things like have zoom in, zoom outable maps to show where localities are as the genomic data come in and the genomic heat maps come in. We can also zoom in on those in various different ways. This is just sort of one example with a few screenshots that Ryan put together for us of our current sampling. You can zoom in on what we have right now on that map. You can continue zooming in. And if you wanted to see where, where a Scleroporus occidentalis for Berkeley, exactly what that dot is and, who's responsible for it. In this case, it happens to be me. You can do that. And every one of those dots has those metadata associated with it so people can learn about that. Looking forward, we have, and again, I think an email on this is going, maybe it already went out. I don't remember if it went out today or if it's going out tomorrow. But we're going to have a sort of 2-hour webinar discussion that will be running on both December 7th and 10th to really try to nail down a bunch of complicated issues with resequencing and making sure that all of the data are collated and collected in an intelligent way. Give people, we have the mini core at UCLA is now up and running. We just purchased a liquid handling robot and we're getting that programmed up on Monday to be able to start moving people's samples through. Yesterday late, we sealed a deal with UC Davis to buy, Davis is buying it, but we're going to be using it a lot, a new PacBio Sequel 2E, that's the machine that does hi-fi long read sequencing, that'll be exclusively for the CCGP for the next year. So that'll allow us to push all those reference genomes through quickly. And we're developing pipelines for those common analyses that we want all the data to go through. And this is something that we're just starting to circle our wagons around right now, but we're going to need to hire probably a couple of postdocs to help with that. So if you're a good informatician and a great landscape genomicist and you care about conservation, get in touch, okay? thanks, funding, lots of people. But here, the big funding, and again, this is one that I just like to emphasize, is that this is funding directly from the state of California. This is not from the California Department of Fish and Wildlife. It certainly is not from NSF. This was something that a member of the state legislature decided to take on for himself as something that he felt would be important and meaningful as a collaborative effort across the UCs. I can't appreciate that enough. I also can say if you think about projects like this that have really large scope and really large impact across the state, think about getting in touch with your state legislature. Sometimes it actually works out. So I think that's all I have to say. I'll leave it at that. Sorry, it was a little bit rushed, but and I don't know how many people are even still left on the call, but hopefully a few.
Yeah.
And
let's thank Brad, and if everybody can unmute and clap. Yay. Okay.
Okay.
There's still quite a few people. Hi, Brad. Thanks.
Hi. Yeah, my pleasure, Carol. And I do apologize for the
that's okay.
horrible screw up. I don't know what happened. But anyhow, are there any questions?
And if people have questions, you can either put them in the chat or raise your hand, and I can sort of help field the questions with you, Brad.
Okay.
Let's see.
I'll start. I have a question, Brad. That was fantastic. Really fun to see it all put together like that. But first, just a comment. I mean, in addition to all of the great conservation implications of having data like this, of course, there's a lot of just basic biology and population genetic questions that we can address, and that's one of the things that I'm very excited about. So, as you know, virtually all of the specimens in the projects that we've submitted have virtually all of the samples have vouchered specimens. So I actually think there's some real potential to connect genotype with phenotype in these data, and find, as you called, some of the genes that matter for adaptation, especially if we supplement samples with other individuals in the same area. So I think there's the potential to start to think about things like GWAS on natural populations, which hasn't been done a lot. So, but my question had to do more with the conservation side of things. So you outlined the ways in which we hope that this might inform, these data might inform conservation decisions. I don't have a good sense for the frequency with which those kinds of decisions get made at the state level, if there are particular geographic regions that the state is looking at now and thinking about for corridors or land acquisition. Can you just speak to that in general terms a little bit?
Yeah, a little bit. Sure. So I think the primary way that genetics has impacted conservation up until now has been at the single species level. A species is up for listing. The state or the feds are trying to figure out what to do with that listing. They're trying to think about whether that listing should be uniform across the range. And a good example of that is the foothill yellow-legged frog, Ranoboilii, which was just state listed. And it so happened our lab, led by Evan McCartney-Melstead, and also another lab at UC Davis sort of simultaneously did grad-seq studies from across the range of the species, and fortunately came up with identical results. So just wonderful, right? 2 independent studies. And based on that, Laura Patterson, who's the state herpetologist who was dealing with that listing, recommended that of the 5 or 6, depending on how you care to look at it, sort of primary management units across the range of the species, that 2 of them be listed as endangered, 2 of them be listed as threatened, and 1 of them, which accounts for every foothill yellow-legged frog from the Bay Area north, not be listed at all. Okay, so that and that kind of splitting up of the species in that way at the state listing level has only happened 1 other time in California. It was with wolverines. And otherwise, that's not how the state does it. It's an all or nothing thing. And Laura took a big sort of step out into the unknown with that. And I think there will be other examples of that. At the federal level, they do that with distinct population segments, DPSs. And those, when tiger salamanders were listed in California, that was as 3 different distinct population segments. So there's more of a history of it at the federal level, but not at the state level. So that's been the traditional way to do it. But the state has, certainly members of the state have told me in no uncertain terms, what they'd like to be able to do is to do that not at the individual species level, but more at the landscape level. And, like the Wildlife Conservation Board that I mentioned, they spend between $100 and $200 million a year purchasing land for conservation in the state of California. And they do it year in and year out. And they want, good, solid science helping them make decisions. It's not all just about conservation. It's about opportunity and serendipity. But it's also, if there are choices to be made and, one area harbors tremendous amounts of genetic diversity and another area doesn't, they've certainly told me they would like to be able to take that into account. But they don't want to do it just for one species or another. And so I think that, there's an open mind and an open ear to using this kind of information. I don't think there are particular landscapes that are under consideration necessarily, because so much of that is serendipity. But I think there is an interest, like a really big interest, in using these kinds of data to help make intelligent decisions. And the fact that it's techie and the fact that no one else is doing it are both very, very appealing, at least to the folks I've talked to. And that's right up to the highest level, certainly in the Department of Fish and Wildlife, as well as on the federal side. So there's some answer at least. Any others? Other questions, other thoughts?
Sure, I had a question, which is not all of the species that have been selected for inclusion in the CCGP, or many of them aren't even necessarily of conservation concern. So I was wondering, it seems to me that perhaps within taxa species were chosen both perhaps because they were of conservation concern, or then other species have been selected for inclusion because they represent broad coverage across ecoregions. And I get the impression from kind of the coverage maps you were showing based on marine and things, that's how things were done. So I was wondering if you could talk a little bit about that. And
sure.
Data can be differently either for those broad coverage species versus the ones that were included for because they were narrow endemics. Or...
Yeah, so it's a great question, Emma. And so the answer goes something like this. To my mind, the ideal species would be one that is broadly distributed across the state. So it's hitting lots of ecoregions so that when we develop that heat map of genetic variation for that species, we're covering a lot of different parts of the state, a lot of different ecological parts as well as just geographical parts. But also that the regulatory community and the conservation community cares about. And, as you know, usually those two are at odds because most species that are really critically endangered have very narrow distributions, and most species that are very widely distributed are not considered to be in trouble. That's not entirely the case. Western pond turtles, which are now considered to be 2 species, cover most of the state, and they're up for federal listing right now. So there are situations like that but they're relatively, they're not all that common. And so what we said was that a species has to have relatively broad coverage. We're just not going to do, delta smelt that are only in the delta, no matter how important they are, because they're not contributing to that sort of overall species conservation, or excuse me, overall California sort of insights that we really want from this project. So having said that, if there is a part of the species range, if there is a subspecies, so song sparrows are a great example, right? Dirt common species, they're everywhere in the state, but there are a couple of subspecies that are species of special concern in the state of California. And so, when Lori put that one up as a possibility, we said that makes a lot of sense. That's a fine example. Another is striped racers, if you know them, Masticophis lateralis. There's a little narrow endemic in the Bay Area that is listed. Otherwise, it's a big wide-ranging species. When you combine it with the coach whip from the deserts, it becomes a very wide-ranging group of species. So again, you get both the breadth and you get some information on that endangered thing. And that's what we've tried to do. We sort of informally allocated maybe a quarter of the species to ones that had no conservation value, but were in our minds sort of so important ecologically or so widespread that they would tell us so much about variation across the state that we wanted to include them. So things especially like oak trees that are, there's several species of Quercus that are in the study that are not endangered, but they're just these sort of ecological foundational species. Western fence lizards, Clopus occidentalis is another one that it's just everywhere. And so that is a compelling one as well. But we didn't want there to be, we didn't want that to sort of overwhelm the choice of species. And people, when they applied, and this was all, people had to apply, there was both an internal and external evaluation committee that read every one of the proposals that came in. I don't know, about half the proposals were funded, maybe 2/3. So it wasn't all that competitive, but if species didn't fall into those categories and didn't make sense from one of those two perspectives, then they weren't funded. And we did, as people like Michael and Rory know, we also did some horse trading to say, well, how's about we swap out this sparrow and we stick in this one instead? And that was also part of the game to try to sort of maximize the opportunities to get both conservation and foundational species in there. The only other piece that went into it is, and this was really more on the marine side than anything else, is we worked very closely with Mark Gold, who's the head of sort of ocean protection in Gavin Newsom's office, to say from a sort of fisheries management point of view, what are species that you think just have to be in this? And some of those are very declining species like abalone or certain sea stars. Others are just exploited species like Dungeness crab or halibut. We made sure, to the extent that we could, that we included those as well, so that it also sort of has some relevance to this sort of commercial take of species and how populations of those species are doing and what they might consider from a fisheries point of view. So there's an answer for you.
Yeah, thanks very much. That's very interesting. And thanks so much for leading this effort as well.
Yeah, that's been really interesting. That's for sure. Any others? I know we're getting pretty late here on time, but anybody else? Yep. Hey.
I'm really excited about the CGP for a number of reasons, and congratulations on getting the funds for it. It's really a cool project. I just have a question out of curiosity. my field's not conservation genetics, so maybe I'm off point here, but I have seen some papers, like a recent one that came out that was really surprising to me, showed that low genetic diversity is not necessarily a predictor of decline, and that sometimes reintroduction efforts you actually want to take from the low diversity populations because they contain the specific alleles that would allow them to survive. So I'm curious kind of how the CCGP is going to, like, are there plans to address this mismatch, potential mismatch between population structure and conservation concern?
Yeah, well, again, it's a good question. And of course, that's part of the reason why doing whole genomes makes sense, right? Is that if there are sort of weird outlier loci that are, that show very, very high FSTs across habitats, whereas other ones don't so much, those become candidates for genes that matter in terms of local adaptation, and one might want to think about that if one's doing repatriation or other sorts of things. I think, though, I think the general rule that populations that harbor more genetic variation or individuals that harbor more genetic variation in general are more resilient is a pretty bomb-proof rule. It's not absolutely always the case. It certainly isn't. And certainly if there's very, very strong directional or stabilizing selection on particular alleles in particular habitats, that won't be the case for those alleles in those habitats. But you can talk about sort of generalities, or you can also dial in on very specific cases. And I think, the more we all wear our sort of basic evolutionary biology population genetics hats, the more we oftentimes focus in on those individual genes and individual landscapes that we find fascinating, because we're interested in local adaptation, we're interested in whatever else it might be. But I think if you're saying to the Nature Conservancy, you have a choice between buying the Dangermont Preserve, which they just purchased, or Tahone Ranch, and they're both giant pieces of real estate, and you've got to try to figure out from the perspective of TNC's mission, which might be a better investment. I think there, you sort of want a picture of those landscapes. You want a picture of it in terms of alpha, just what species richness look like. You want a picture of population health within those species. You want a picture of, as best as you can predict it, something about resiliency. And I think that's where, a project like this maybe kind of kicks in a little more. And as Michael said, I mean, the thing about it is that all those data are going to be collected. All of those data are going to go back to PIs, right? The reference genomes, the resequencing data there, and we can all nerd away to our heart's content on trying to understand, coat color genes in mice, or why some pond turtles are dark and some pond turtles are light, and what does that mean in terms of the world of pond turtles. And that's great, and I think that's fabulous, and I hope tons of that happens. We're also going to pipe it all through a single set of analysis pipelines so that it can all be combined, so that we're using the same filters on all the data, so that we're setting the same thresholds on those filters as much as possible, so that we have combinable data that we can use to answer that sort of TNC question or whatever. So anyhow, I certainly agree, sometimes, simulation studies and empirical ones have shown that, sometimes in some situations, genetic diversity isn't the issue. Island foxes are, essentially flatlined out on St. Nicholas Island, right? But that's a real exception, it's really not the normal case. At least that's my impression. Okay.
Thank you.
Sure. Anybody else?
it's 1:30.
True.
So let's just give anybody who wants to ask a last question an opportunity to do so in the chat or by unmuting yourself.
Wow, I see Mark Stromberg and Kay Yana both on the line. That's great. Hello to both of you. Good to see you. Hey, last questions? No last questions. Okay, well, sorry for the rocky start and for zooming through it, but at least it gives you a little sense of where we're at with the project right now.
That
Thanks
was
very much, Brad.
Thank you so much for doing that, and thanks for spearheading this whole CCGP. It's wonderful.
Yeah, well, it's let me tell you, the only other word of advice I'll give is sometimes be careful what you wish for. You know, I've been wishing for this for quite a while, and it's great, but it's a very interesting exercise. It's just very interesting. So, but it does, you know, I think it also is